All right, thank you to everyone that's joining us today, and thank you in particular to our presenters today. Jonathan Watts, who is a scientist at University of Massachusetts Medical School. He's an associate professor at the RNA Therapeutics Institute, and he is working on several Rett syndrome projects that he and his colleagues are going to present to us today. And Jonathan, I'm going to turn it over to you and let you introduce your colleagues and say a little bit about yourselves as well.
Great. I'm going to unshare my screen and we'll tell you a little more about ourselves. So this project that we're going to tell you about today is focused on DNA editing. We want to really introduce the ways that DNA editing can be used for treating Rett syndrome and the pieces that we're working on within that. I have two graduate students who are going to be presenting pieces of the work with me as well, Kevin and Karthik. They're both students in Scot Wolfe's lab. And this project is much broader than just my lab. This is a collaboration involving 4 labs at the medical school. The labs of Eric Sontheimer, Anastasia Korova, Scot Wolfe, and my own lab. And so I'm going to stop sharing for a second and invite Kevin, why don't you go first and just share a minute or two about your journey to grad school and to work on Rett syndrome in particular.
Thanks, Jonathan. So my journey to grad school and pursuing a PhD really started at a really young age. Both of my parents suffer from lifelong debilitating diseases, my dad being a kidney transplant recipient and my mom a survivor of ovarian cancer. And so growing up, I idolized doctors, nurses, and scientists who really took time and effort to teach me about everything that they were doing and for helping my parents. And so when I went to college, I decided to pursue biology and chemistry. And as I was approaching graduation, I had a fight with the dilemma of pursuing medical school or getting my PhD. But at some point during my senior year, something really clicked when I was working in the lab of Dr. Jennifer Bath. I found myself really drawn to the problem solving and the curiosity behind scientific research. And also I realized that this allowed me to not only interact with people one-on-one, but also a lot of the things that I did could solve problems on a very macro and large scale level. And that this led me to grad school. And obviously in our grad school, we have many opportunities to do rotations. And one of them I did with Scott, and I immediately fell in love with the science of DNA editing and how a lot of the things that we can do with this technology can be everlasting and can help a very large community of people, including, Rett syndrome itself.
Awesome, thanks, Kevin. Karthik, can you share a minute about your journey too?
Yeah, sure. Hi, everyone. I'm Karthik, and I grew up in India, where I spent probably the first 21 years growing up there. And then I decided that I should leave this warm tropical weather and move to Boston to get my master's. So what got me interested in science, if I look back, I would probably say my mom. So she has a master's in chemistry, and she was in a way my first science teacher. And growing up, chemistry was always my favorite subject. But that was up until the 7th or 8th grade, where I finally learned what DNA is. And once I learned that, I was just super curious to know more. I started reading more about DNA, trying to find out how it's packaged in a cell, how it functions, how it repairs itself. And basically, I was so obsessed that I would end up finishing the biology textbooks within the first few months of the academic year. And that's how I knew after high school I wanted to pursue this further. So I got my undergrad degree in biotechnology. Then I got my master's in molecular and cell biology from Brandeis University in Waltham. And at Brandeis, for the first few months, I was working on DNA damage and repair pathways and bacteria. And so when I graduated, I worked at Dana-Farber Cancer Institute. And again, I was working on DNA damage and repair pathways there, but this time in human cells related to a disease called Fanconi anemia. So during those 2 years at Dana-Farber, I finally realized that what actually drives me and motivates me to work every day is not just to find answers to these unknown questions, but to put out some work which would directly impact patient healthcare. So when I decided to get my PhD, I wanted to work in a field which would have a much more direct impact on the society. So it was no surprise that I, as well as like others here, jumped on the CRISPR-Cas9 bandwagon, and I joined Scot Wolfe's lab, and it's been 3 years now. And I would say I'm just getting started. I'm happy that I'm working on the Rett Syndrome project here, and I'm glad that I have an opportunity to talk to you guys today.
Thank you.
Yeah, thanks very much, Karthik. As Monica said, my name is John Watts, and I'm really delighted to be able to share with you guys today. My background also is in chemistry, and so I really have come to be passionate about applying chemistry to the development of platform technology to treat disease, including rare disease. So as Monica said, we actually have really been grateful for the support of RSRT for several projects at UMass. And today we're going to focus our presentation on projects specific to DNA editing.
The RNA Therapeutics Institute at UMass is really an amazing assembly of just top-notch labs. And, the fact that you get to interact and to brainstorm with people like Eric and Anastasia and Scott, you don't get that opportunity at every institution, right? It's really something special.
Absolutely, it's just amazing. I actually started my independent academic career as a professor in England at the University of Southampton in a chemistry department. And 5.5 years ago, got an invitation to move with my research group here to the RTI, the RNA Therapeutics Institute. And it's been just so much fun. And it allows us to really work across the whole spectrum, because the RTI brings people like me who think primarily about chemistry, or at least I did before I moved here, with then physicians and RNA biologists and structural experts and gene editing experts like Scott and Eric. And so it's been a real privilege, and it certainly is a fun place to be.
And then the only other thing I'll mention is if you have questions, to please put them in the Q&A section, which you should see at the bottom of your screen, and we'll try to address those at the end. And if there's any questions that we can address, I'll do my best to get answers and email them to the people that ask them. Okay.
Thanks, Monica. That's great.
Let's get started.
All right. So to understand the context for gene editing and how DNA editing or gene editing fits into the treatment of Rett, we need to go back again to the central dogma of molecular biology. Fortunately, there is a webinar on this topic on the RSRT YouTube channel. And so you can review that, how the central dogma works and how it fits into Rett syndrome specifically. And it's really well done. I'm actually really impressed with the resources that RSRT has put together. So as you know, as those of you who've watched that or who know from other places know, Rett syndrome results from mutation in MECP2. And a mutation in the DNA, of course, propagates to the RNA, which propagates to a mutant protein or a truncated protein. And we can address the disease by targeting, in any of those 3 levels. We can intervene to correct the problem at those steps. And so this is taken from the RSRT website, focusing on these kind of 6 areas of therapeutic curative intervention. And one of those is focused on the protein level. 2 of those are focused on the RNA level. And again, another shout out to a previous webinar in this series on RNA editing that Gail Mandel and John Cinnamon put together. That's again really well done on this kind of lower right quadrant. And today we're talking about DNA focused approaches. There are 3 of those. And we're focused on gene editing among those. And so let's think how let's just look together for a minute how these 3 DNA focused approaches differ from each other. So gene replacement or gene therapy is of course introducing a separate copy of MECP2 without the mutation in it. And we're all very excitedly sort of holding our breath for this Novartis Avexis trial, hoping for the best in that. The second approach, MECP2 reactivation, is based on the idea that Rett patients, female patients, have a second copy of the X chromosome with a correct version of MECP2. And if we can find a way to turn that chromosome back on, we can use the copy of MECP2 that's found on that newly activated X chromosome. And then the approach that's the subject of today's webinar, gene editing, seeks to bring in a protein RNA complex that finds the mutation and corrects it in the endogenous copy, the main copy that contained the mutation before. And so all of these approaches are a way of addressing the problem at its root, but each of them with a slightly different twist on how to do that. And again, just speaking to the amazing strength of this community, RSRT is funding multiple groups in all of these areas. And so it's really quite exciting to see how this is moving forward. So today we want to talk about 3 approaches that are variations on CRISPR technology to treat Rett syndrome by editing DNA. So I don't know, I think we've heard a lot about CRISPR. I don't know how many of you know where the name comes from or where CRISPR came from. CRISPR is actually based on a bacterial immune system. We've thought a lot about viral pandemics this year. And I know that many people have really suffered very deeply with this pandemic. Bacteria are constantly under assault from their own viral pandemics. I don't know if that's something that you knew, but every second, phages carry out an estimated trillion successful attacks. Phages are viruses that attack bacteria. And they way outnumber bacteria in the world. They are incredibly destructive to bacteria. They kill about 40% of the bacteria in the ocean every day. Unfortunately, bacteria also multiply really quickly, so they can keep up. But they really are everywhere. A teaspoon of seawater contains as many phages as there are people in my home country of Canada, 35 million or so phages in a teaspoon of seawater. And so bacteria have to protect themselves if they're going to have a chance of surviving their own viral pandemic that's ongoing day by day. So CRISPR is an aspect of a bacterial immune system, and it's named for a series of repeats, clustered, regularly interspaced, short palindromic repeats. It was identified in sequencing data, DNA sequences, before anything else was known about it. It was this kind of curiosity in sequences. Those little black boxes are repeated sequence, the same sequence every time. And then the little diamonds of different colors are different spacer sequences, different sequences of DNA that come intervening between those black boxes. And then there are a series of proteins that were always found near this repeated locus, and they were called CRISPR-associated proteins, or you might have heard of people talking about Cas9 or Cas proteins. And that's what that is. That biggest protein in this case is Cas9. And so when we talk about CRISPR, we're really making a reference to this repeat structure. And a key insight was from the Spanish biologist Francisco Mojica. And Mojica observed that the spacers were based on sequences from phages or other kinds of invading DNA. And more importantly, when a bacterium contains a spacer that matches DNA from a particular phage, it's immune from infection by that phage. And over time, it was observed that bacteria, if they managed to survive this viral attack, they could incorporate a little bit of the phage's DNA to grow its array of CRISPR repeats and protect itself and its progeny from future attacks by that phage. So how do we go from an immune system in bacteria to gene editing for Rett syndrome? Well, again, a number of key insights along the way. One of them from our collaborator and colleague, Eric Sontheimer. He demonstrated in 2008 that this bacterial immune system, CRISPR, works by recognizing and cutting invading DNA. So it's a system that cuts DNA at a particular sequence. And then Jennifer Doudna and Emmanuel Charpentier, whose names, again, you may recognize since they were awarded the Nobel Prize just a few months ago for this discovery, they showed that this same CRISPR system can be reprogrammed to cut DNA from other species. And so here's what it looks like. This is Cas9 protein in the blue sort of oval. We have an RNA guide in this dark blue, and then a yellow, light blue piece of RNA as well. And that dark blue section is what came from those spacer sequences in the CRISPR repeat. And that's what remembers which DNA to cut, whether it's DNA from a phage or whether we're using it to cut DNA relevant to treating a disease. And the gray here is target DNA. So it opens up the duplex of DNA, comes in base pairs, and you can see the little pairs of scissors that are able to cut DNA. Those are two different domains of the protein that in its native form cut both strands of DNA. So why is this so important? It's pretty simple in many ways, this idea that we can use a protein RNA complex to cut DNA. But an analogy is helpful here as to why this has been such a revolution. There was technology prior to CRISPR for cutting DNA, but it required a different protein for each DNA sequence. And so it would be like needing a new computer for each program that you wanted to run. You'd have to open up one laptop to write an email, and a separate laptop to make a presentation, and a separate laptop to watch YouTube. Obviously, this is really limiting in terms of our pace of being able to do things. But with RNA guides, those guides can act like software. The same protein can be taught to cut any DNA sequence by giving it the appropriate RNA guide. So this greatly simplifies the task of cutting DNA. So how does cutting DNA lead to a therapeutic benefit, lead to an edit that can be curative? Well, when the DNA is cut, the cell does a pretty good job of repairing it. Often, though, the repair includes small errors, insertions or deletions. We call them indels for short. And for some kinds of diseases, it's useful to introduce indels. These have the effect of inactivating the gene. So there's a drug in clinical trials right now that inactivates a protein called TTR to treat TTR amyloidosis. It just cuts, and then when the cell repairs, it inactivates the protein. If it makes a perfect repair, the Cas9 can cut again until these indels take their place in the gene and inactivate expression of the gene. But that's not particularly useful for us for treating Rett syndrome. There's a second pathway. In dividing cells, we can give the cell a piece of homologous DNA with some shared sequence, and it can use that homologous DNA as a repair template. But this homologous recombination doesn't work very well in neurons. So that's still not going to be a great option for us for treating Rett syndrome. And so today we're going to talk about three approaches that do have real potential to be useful in Rett syndrome. First, I'll explain a little bit about exon replacement by targeted insertion. And then Kevin will walk us through a technology called base editing, and Karthik will explain a bit about something called prime editing. The MECP2 gene contains 4 exons. So if this was a long stretch of DNA, the parts that code for protein are quite short. And as the DNA is copied into RNA, those exons are spliced together to make one connected piece of RNA, which is much shorter than the DNA that it was copied from. This dark blue part is called the UTR, or untranslated region. And the light blue part is what codes for the MECP2 protein. And most Rett disease-causing mutations, of course, are found in exon 4. And so one approach using homology-independent targeted insertion is to synthesize a version of exon 4, an artificial exon 4, and we would use a miniature shortened version of the UTR, similar to what's used in gene therapy approaches. We could insert it just upstream of the patient's exon 4. And if there's a few changes in the frame or a few nucleotides plus or minus just before or after this, it doesn't make so much difference because this would be in an intron. And so we would add a strong splicing site here so that it would signal to the splice machinery to use this exon 4 instead of the old exon 4 to replace the mutation with a correct copy of exon 4. You could imagine a similar approach for exon 3, where we might be able to combine an exon 3/4 artificial construct containing both of those exons and a miniature UTR, again, just upstream of the patient's exon 3. The way that this is done in homology independent targeted insertion is to provide this short piece of DNA with a cleavage site that Cas9 can recognize, the CRISPR machinery can recognize and cleave. And we put that same cutting site as the site in the patient's DNA, except we put it in the opposite orientation. And that way, if the insert goes in upside down, it looks like the insert did go in upside down there, Cas9 will cut it again until the insert comes into the right orientation, at which point CRISPR can no longer recognize the cut sites. So that drives the equilibrium toward the desired product with the mutation going in, the insert going in the right orientation. So there's a real advantage, a real attractiveness about this approach because one intervention could be useful for the majority of patients. The real challenge of this approach is that so far it works with relatively low efficiency. We have in our labs observed some of the correct product, but so far a lot of other editing outcomes have been competing with that product. So simpler repair outcomes where the cell introduces insertions, small insertions or deletions at the target site, which CRISPR can't then re-cleave, can be quite competitive with the
you mean
process.
you mean off-target edits, right?
Well, it can be cutting at the right site, but instead of driving repair by using that piece of inserted DNA, it can just repair between the two cut ends of DNA. And if it repairs that in a way that it doesn't allow the CRISPR complex to cut again, then that's sort of a dead end for editing. It's not able to be cut, a second time to re-allow the insertion of this artificial exon 4 or exon 3/4 construct. So there may be ways to address those, but this is so far has proven in our labs to be a challenging approach to drive to the levels of efficiency that we would want to see for clinical use. However, the good news is there are higher efficiency approaches available that we have now seen some pretty high efficiency conversion to the desired product. And so I'm going to pass now the mic and control of the screen to Kevin to tell you about the first of those approaches.
Can I just interrupt and ask a question?
Yeah.
So the challenges that make the efficiency, and by that we mean, right, the number of edits that you're seeing, so you might see in one cell out of 1,000, something like that, right? So is this an engineering problem? And are there technologies and people that are working on this to improve the efficiency? Is that something that 6 months from now, a year from now, we might be able to say, hey, we're getting 50% efficiency?
Yeah, it's a great question. The challenge for an approach like HITI is that we have to have the repair construct, the construct to be inserted, present at the site of cleavage. At the same time that cleavage is happening, and it has to be inserted before the two cut ends can come back together on their own. So there may be ways that we could engineer this complex to pull the inserted construct along with the Cas9 protein, for example, near to the cleavage site. By definition, when we've cut DNA, the two ends of that DNA are close together. And so there's a high chance, relatively high chance, that they'll find each other. And so what we need to do is increase the odds that they'll find the artificial exon that we've introduced before they find each other, at least before they find each other in a way that causes insertions and deletions, these sort of dead end editing outcomes that are then hard for us to recut and reintroduce. So those are the sorts of engineering approaches that we could consider. But I think that anytime we need more things to be in the same space at the same time, statistically, that's a harder problem. It will tend to be harder than having fewer things in the same place at the same time, especially if those things by definition have to be in the same place at that time.
Thank you.
Thanks, it's a great question. All right, over to you, Kevin.
All right, thank you, John. Sorry, this is a little embarrassing, but it seems that I have a typo on my screen. But this will allow me to kind of highlight what base editors are. Let's see. Oop, there we go. Okay, much better. Base editors, as John mentioned, it's a newer tool that allows us to more efficiently edit the DNA. And so this all started a few years ago in David Liu's lab out in the Broad Institute. And while Cas9 itself is able to create double strand breaks, and then with a donor, we're also allowed to integrate or correct sequences pretty efficiently, but this is still suboptimal. You're still creating breaks in the DNA, which can be detrimental in some cases when there's like DNA rearrangement and also DNA repair factors that could be harmful to the cells. So what Dr. Liu and his lab noticed was in this pie chart, they're representing all genetic diseases. And as you can see, a lot of them are just single point mutations. And what they noticed is that creating these double strand breaks, because it's harmful and so many of these mutations are so small, is there a way that we can correct them more efficiently? And to this end, they created what we know as base editors. One specific class of base editors is adenine-based base editors, which is a mutant version of Cas9, where it is unable to create a double strand break. Instead, it'll nick one strand of the DNA, and it's fused to two proteins, which allow it to specifically change a single A residue in the DNA. It is deaminated from an A to an inosine, which when read by DNA polymerase, when it comes through to repair the DNA, is read as a G. So basically the take-home message is that adenine base editors allow us to convert, allow us to convert A's to G's, or because DNA is complementary, allows us to convert T's to C's on the opposite strand. And so with this tool, what we realized was that actually when we dive deeper into these point mutations, over almost half of genetic diseases can be corrected if based on this blue circle here, as long as you can convert an A to a G. And also, yeah, so a majority of genetic disease can be corrected using these admin-based editors, which is why it makes it such an attractive target site for us in the gene editing field. And so specifically, when we look at mutations associated with Rett syndrome in MECP2, here I'm just going to highlight one specific mutation. This is the 763 C to T mutation, which creates a stop codon here, highlighted in red. So this mutation creates a stop codon leading to decay of the protein. However, theoretically, when we treat with ABEs, we can revert that A to a G, thus reverting back to a normal arginine base amino acid. And so how we look at this, here I'm showing a Sanger sequencing chromatogram, which each peak just basically represents a base with colors associated with ATCG. And here I'm highlighting this specific 763 mutation in these patient-derived fibroblast lines. So they carry specifically the CD2 mutation as well as the wild type allele, since it's heterozygous. And following treatment, you can see instead of, if you kind of squint a little, you can see the disappearance of the red bump, and it becomes majority this blue peak here, representing a corrected allele. And so this is how we in our lab look for base editing specifically. And so in our labs, we've applied this technology to several common MECP2 mutations. As I mentioned before, the 763 C to T, as well as 808 C to T, and 916 C to T, all of which are mutations in the MECP2 gene that that have a fairly high frequency fairly high frequency of greater than 5%. And so just kind of going over our experimental design, we obtained patient-derived fibroblasts from the Rett Foundation. And we treat the cells with the Cas9 AB ribonucleoproteins, which are the recombinant proteins themselves, complex to the RNA. And we deliver these to the fibroblasts by electroporation, which delivers a small electric pulse to the cells, allowing the protein to be imported into the cell. And from there, it can be shuffled into the nucleus where it can do its job. And then after 72 hours or so, we can look at editing in these cells. So here, starting off with the first mutation that I talked about, the 763C to T. Here I'm highlighting the sequence in which we're targeting. In red is the specific A base that we're targeting. And to my right is the quantification of the Sanger sequencing that I showed before, the difference going from the control to the treated sample. As you can see here, in these primary patient lines, we're achieving very significant editing of greater than 70% at this mutation. This is something that's really never been seen before, at least in these cells targeting these mutations. So moving ahead, we also, as I mentioned, tested a few other ones, 808. Again, doing phenomenally here, we're achieving greater than 70% editing again. And lastly, 916, which is the last mutation that I'm targeting, is also greater than 80%, or greater than 70%, sorry, in overall conversion from A to G into the wild type sequence. So moving forward, there's obviously a lot of steps until this gets into patients, but moving forward, we need to test these proteins in vivo. By this, I mean in model systems like mice, so that we can make sure that this is still efficacious in a living organism, and also that it's safe and safe and tolerable for other organisms besides cells. And this is obviously a even more critical part is to really quantify off-target effects related to base editing. And by this, I mean like editing at undesirable positions, or editing RNA. These are things that the community is very cognizant about, since this is such a new technology. These are things that we really focus on in our labs and other labs when we use these tools for disease treatment. And while base editing is a phenomenal technology, and it's growing very rapidly, especially with the advancements in our labs and other labs like David Liu's lab, there's still some limitations. One of them is bystander effects. By this, I mean, here I'm using another common Rett mutation, this threonine 158-2 methionine mutation. Here I'm highlighting this specific A mutation that we want to target. However, a current issue or limitation with base editing is that we can't just target that one base. There is kind of a window of approximately 5 to 10 bases that the base editor can function. So if there are other A's surrounding our desired mutation, there's a possibility that we may change those bases. So we're looking at ways of either shortening the window or having technologies that are more specific in terms of what they're trying to change, which is what Karthik will be talking about. And obviously, as impressive as base editing is, especially in cell lines, it does require a separate intervention for each mutation. There is no specific silver bullet to target all the mutations commonly associated with Rett. Unlike John's technology that he talked about, or what Karthik will be talking about next, base editing is very specific. That's kind of what makes it special. However, again, it requires us to have separate interventions for each mutation associated with it. So from here, I guess I will turn it to Karthik, who will talk about prime editing.
Yep. So I shall neither confirm nor deny that we spent the last 24 hours proofreading our presentation, because we just don't seem to be able to get rid of these typos here. John, if you could give me the access to move the presentation forward.
I think you should have it, Karthik, see if you can click through. There you go.
Yeah. So we have a typo here again, but luckily we have more tools to help us. I don't have to rely on Kevin's base editor here. I can use a new tool called Prime Editor that I'm going to describe in a minute. It can do the same job what the base editor did, which is change a single character to another character. In the meantime, I also want to highlight what else it could do, like what other additional edits it can create when compared to base editors. So if you see here, this is my full name. And if you remember in the beginning, I introduced myself as Karthik. And there was a reason for that because my full name is Kartiks Ponnienselvan and it has 9 syllables. And usually I love watching people struggle to pronounce it, but at times it's really painful. So I would like to delete some characters here. And the question is, can prime editors do that? And similarly, if I want to introduce more characters to make this more descriptive, that's UMMS, that stands for UMass Medical School, can prime editors introduce more characters there? And the answer is yes. So you can see how versatile this tool is. It can not just change single characters, but also introduce and delete characters. And I wanted to emphasize that point because this tool can not only be used for the T158M mutation that Kevin was talking about, where base editors fall short because of the bystander adenines on the side here. It can also be used to target other MECP2 point mutations. So how does this tool work? It's a fairly new tool, again, developed by David Du's lab at Harvard University. And it has 3 main components. A Cas9 nickase, which cuts a single strand of DNA, and a guide RNA that's been modified at its distal end. It's been extended to contain the information that has to be copied at the DNA target site. And this modified guide RNA is called as pegRNA, which stands for prime editing guide RNA. And the final component of this tool is a reverse transcriptase, which essentially is the main component of this tool, which copies the information from the RNA to the DNA target site. So I'll walk you step by step how this tool functions. So first, the Cas9 nickase cuts a DNA strand and sort of liberates a free DNA strand here. And the prime editing guide RNA sticks to the free DNA end or binds to it. And once it's bound, the reverse transcriptase then copies the information from the pegRNA to the DNA target site. And the edit is highlighted in red here. So once the edit is incorporated at the target site, after that, through several rounds of DNA repair, the edited DNA is incorporated at the target site. So that's how this tool works. And since we have described different tools to you, you guys might be wondering how we deliver it to brain or brain cells to carry out the edit there. So that brings me back to the central dogma of molecular biology that John explained in the beginning. We can deliver these tools as DNA, RNA, or protein to the target cell. But eventually, it's the protein that carries out the editing function. So if we supply the cells with DNA or RNA, they have to be eventually translated into a protein to carry out the editing function. And the means of delivery, we can classify them mainly into viral or non-viral means. The AAV method, a lot of the members in the audience might be already familiar about it because AAV is used in gene therapy to provide a functional gene copy to the target cell, but we can at the same time use this AAV viral envelope to package a DNA that codes for the editing tool and then use that to deliver it to the target cell. The non-viral means can be further classified into physical and chemical methods of delivery. So physical methods are pretty straightforward, where we can just micro-inject these DNA, RNA, or protein form of these editing tools directly to the cell. Or we can electroporate these cells, basically zapping them with low voltage to open up the pores in the cells so that these DNA, RNA, or protein can sneak inside the cell and carry out the editing activity. But as you can imagine, these physical methods are not going to be a suitable mode of delivery for the brain. And we would have to rely on AAV or chemical methods for that. And to briefly explain the chemical methods, we fuse the Cas9 with a chemical sequence or kind of coat them with a chemical formulation to facilitate their uptake by the target cell. But in our lab, in the Wolfe Lab, for the preliminary testing, we always test them in cells first. We test our tools in cells using the electroporation method. And protein is the favorite form of the editing tool that we like to use. And the reason for that is because we have seen in our lab with base editors and other tools, when we use a protein form of the tool, we tend to see a higher editing activity compared to the DNA form of the tool. And you can see here, when we use a small amount of the base editor protein, we are able to achieve around 50% of the editing activity, meaning 50% of the cells are corrected. And when we use the DNA, it's around 14 or 15%, which is why we prefer using protein, and which is why I want to try and make the prime editor protein. So I just want to just give a quick rundown of how we make the protein in the lab. We first insert the DNA in the bacteria, the DNA that codes for the editing tool that we are interested in. In this case, the prime editor. We introduce it into the bacteria and then select the bacteria that has the DNA and culture them overnight in a soup of nutrients. And then the next morning, we collect the bacteria, break them open, and extract the protein, run them through a series of column purification, which at every step separates our desired protein from other impurities, and finally leaving us with our desired purified protein. After we have that, we want to make sure that what we have finally purified is the protein we are looking for. So to do that, visually, we run it on a gel that separates protein based on its mass, charge, and size. And you can see here in the last 3 lanes, the prime editor protein, the band for the prime editor protein is clear and strong. Whereas on the left side, when we don't run these proteins through the column purification, you can see there are several other proteins that are produced in the bacteria that come along with it. But when we run it through the column, it's separated out from the other proteins that are present in the bacteria. So now that we have purified prime editor protein, the next step that we want to do is do a series of quality control tests to make sure that the prime editor protein we have is functional, and then test them in patient fibroblasts, specifically the T158M mutation, and just target that mutation and try and fix it with the prime editor, and then move on to other mutations as well. And then finally test them in vivo and mice. And hopefully next time, if I get a chance to talk to you guys, I will have an update on how this all went. Now, to end this talk, we would like to give you guys something to think about and be hopeful about, and I'll give it over to John.
Thanks, Karthik. That's great. So, I've seen coming up in the questions, questions like how long until one of these is actually available in patients, and I think that's a question that we all are working toward and want to know the answer to. And we thought it would be helpful to give a sense of what gene editing, what the progress is in gene editing for other diseases in terms of real use in patients in the clinic. And you can see this is pretty recent. This is something that's made huge progress just over the past couple of years. So the first patients were cured from blood cell diseases like sickle cell disease, sickle cell anemia, and beta thalassemia in trials that initiated in 2019. There are also multiple approaches where blood cells are being engineered in a way that allows the immune system to be programmed to recognize and destroy cancer cells. You saw Kevin present data where he electroporated fibroblasts, and it was very efficient. And the advantage of blood cells is, of course, they can be taken out of the patient and electroporated or treated with some other high efficiency delivery approach. And so those are a very natural starting point for gene editing. The eye is another tissue where there's significant progress, and that's now, instead of pulling blood cells out and editing them, that's editing, inside somebody's eye, obviously. And so there was a clinical trial for a kind of genetic blindness called LCA10, Leber's congenital is what that stands for. And then that was initiated just last year. And also in the liver, a genetic amyloidosis, TTR amyloidosis, initiated by Intellia just last year. So there are reasons to be very encouraged. So far, there is not a clinical trial in the brain using gene editing. But that is something that we are working to change. I want to just show you a couple of slides of evidence that we can do in vivo gene editing in the brain. These are slides made from a section of the brain of a mouse done in our groups just in the past couple of months. The same 4 investigators, Eric, Anastasia, Scott, and myself, have funding through the NIH to work specifically on delivery of gene editing machinery to the brain. And so what I'm showing you on these slides is 2 different types of delivery. The mouse on the left has had an injection into the striatum of its brain. This is this sort of central section, if you can see my mouse, of a Cas9 protein with a chemically modified guide RNA. And you can see along near the site of injection, there's really substantial editing. This mouse has a fluorescent protein that it expresses when editing has occurred, and that shows up brown in the slide based on the way that we're viewing those cells through the microscope. On the right, you can see a very different way. That's using a viral vector, an AAV vector, to provide the gene editing machinery. And that vector then has the Cas9 DNA, which is transcribed into RNA and translated into protein, and then carries out the editing. And you can see there's now a less concentrated, focused area of editing, but editing pretty broadly through both sides of the brain. There are a number of edited cells there. So this is work that's very much still a work in progress. We're pushing forward to improve this day by day and week by week, I think, moving it forward. These 2 pieces of data are just in the past 6 weeks. We've had both of these pieces of data. So, we're excited to share that the in vivo delivery to the brain is moving forward. And other groups besides our groups are also working on this. One of the really exciting things is that the progress we make on these Rett mutations and technologies can also be combined with synergies from other groups' contributions. And one of the things, again, that I want to highlight is that the group of Guoping Feng at MIT has just made a really important, really useful mouse model with RSRT funding that we can now use to test these approaches on the human sequence in the brain of a mouse. And so, again, we're just really grateful for the support of RSRT to us, but also for the support of RSRT to others in the community who we can work with to move these things forward.
Thank you.
Well, thanks. Yeah, thanks very much, guys. We welcome your questions.
Great, thank you. And I'll just add that this is such an active area, right? So many different labs working on it, and hopefully, we can piggyback on progress. Okay, let's take some of the questions. So I'll start with a question from Laurent Villard. He's a scientist, actually, in France. So he says, is this technology working in non-dividing cells? have you sequenced the DNA of your transfected fibroblasts to estimate off-target effects in vitro?
So both base editing and prime editing are expected to work in non-dividing cells, and there's some evidence that they do work in non-dividing cells. That's the real advantage of these base editing and prime editing approaches compared to homology-directed recombination, which doesn't work very well in non-dividing cells. But actually, all three of the approaches that we talked about today, HITI as well, is designed to work in non-dividing cells as well. So that's why we focused on those approaches. Yeah.
Okay, here's a question from Paul. It would seem that correcting a genetic deletion may be easier than fixing a mutation because a deletion does not require the removal of a mutation. Actually, it does, right? It requires removal of, well, let me finish up. Is that reasoning correct? Is there any research or techniques specifically addressing deletions?
Yeah, right. So prime editing is pretty good at doing mutations, small insertions, or small deletions, up to, I think it's, Karthik, correct me if I'm wrong, I would say about 70 bases are sort of where it's been pushed so far. Is that right? So that's a pretty substantial mutation and might allow one intervention to be used to correct several mutations that are close to each other as well, without having to have a separate intervention for those, because of the fact that it can have a little bit more reach. There's not necessarily something fundamentally easier about correcting a deletion compared to an insertion. As you saw on the slide that Karthik showed, when the reverse transcriptase makes its edit, it does it into one of the strands, and then we count on the DNA repair machinery within the cell, which we sort of bias in a way that we engineer the protein. And this is really the beautiful work of David Liu's lab by designing the protein in this way to convince the repair machinery of the cell to adopt the corrected strand rather than fixing the other strand. So it fixes the non-edited strand to match the corrected strand, whether that's a small deletion or a small insertion or just a base change that keeps the same length.
Okay, here's a question from Dorota. She says, I'm a pediatrician and mom to a 1.5-year-old girl with atypical Rett CDKL5 deficiency disorder. Is there any possibility of treating MECP2 related disorders with these methods? I'll answer that, Jonathan. So, so the same approaches that we're pursuing for MECP2 theoretically can also be pursued for CDKL5 deficiency disorder, but they need their own program, right? They need their own effort, because you can't, the strategies that we're pursuing are fixing specifically mutations in MECP2. You would need the same strategy, but you would need to design programs that are targeting CDKL5. All right, and then a question on why is shorter UTR used for replacement or repair?
Yeah, that's a great question. So essentially, it's because delivering longer pieces of DNA into cells is harder than delivering shorter pieces of DNA into cells. And that UTR of the native MECP2 is so long, it makes that approach really, really quite challenging to work with. And so we're sort of building on the work that's been done by the gene therapy community, but nonetheless introducing that into its endogenous context in the cell so that it has the regulation machinery of the chromosomal context of being built into the chromosome where it's supposed to be in the cell. But it's really just because it's easier to work with shorter DNA than longer DNA.
Great, great. So we're at the hour mark. Most of the other questions have to do with things that we've already addressed, but I'll get in touch with everyone to make sure that their questions have been answered. So thank you so much, Jonathan, Karthik, and Kevin, for joining us. I'm sure I speak for every Rett family out there when I wish you Godspeed in your research, and let's do this again in the not so distant future and bring people an update.
That would be great. Thank you guys so much for your support. And we really do think of you guys as we're in the lab working on these things, and we're excited to be part of this community and really hoping that we can make a difference.
Thank you so much. Bye-bye.
Bye-bye for now.